What's the difference between prokaryotes and eukaryotes? Nobody karyotes. That god-awful joke there pretty much sums up the indifferent attitude of most people towards these microscopic unicellular creatures on Earth. Our modern day society has largely come to associate every prokaryotic organism with one word: bacteria. I mean, it takes just one stroll down the hand sanitizer aisle to see this unwarranted prejudice against bacteria take shape. Antibacterial. Kills 99.99% of bacteria. Helps reduce bacteria that could cause disease. And to think that we complain about our lives. Now, although bacteria often get a bad rap for being disease-inducing organisms, we shouldn't jump to conclusions and regard every prokaryote as inherently harmful because the species that do cause human diseases represent only a small fraction of all prokaryotic species. Further, there also exists a second classification of prokaryotes, the archaea, that has yet to cause a single disease. The genetic diversity among prokaryotes is truly a wonder that we humans, as their multicellular descendants, have long forgotten. And to emphasize the last point, I should note that over 100 trillion bacterial cells have made our body into their home, easily outnumbering all human cells by a factor of 10. Now, for those of you out there already feeling squeamish, don't worry. Most, if not all of these bacteria are mutualists, like the intestinal species currently helping us digest food that our own enzymes could not otherwise. As you can see, we reap many benefits from the metabolic capabilities of prokaryotic species, from bacteria to archaea. For instance, humans have long used bacteria to convert milk into cheese and other dairy products. We've also just recently harnessed the power of thermophilic archaea in the polymerase chain reaction technique, a commonly used laboratory method for amplifying or making millions of copies of a particular DNA region. One specific application of prokaryotes that has intrigued me is their wide use in biopharmaceutics. Bacteria, like E. coli, serve as the backbone of biotechnology. It is through them that we can produce vast amounts of vitamins, antibiotics, and medical hormones like insulin. So, what makes these tiny organisms such great factories? Well, to start things off, unlike their eukaryotic progeny, many bacteria have small circular DNA molecules called plasmids that are replicated separately from its main body of DNA and contain a specific antibiotic resistance gene. These unique features are put to use in gene cloning, one of the foundational laboratory techniques in biotechnology. When you hear the process gene cloning, you may think of the complex next generation cloning of whole organisms like Dolly the sheep. However, the entire process of gene cloning is conducted on a relatively much smaller scale and mostly involves the copying of DNA for purposes like synthesizing antibiotics. In addition, gene cloning itself is pretty straightforward. We first start off with two things: the aforementioned plasmid from a bacteria and a gene of interest from another organism, say a gene coding for insulin. Once the insulin gene is inserted into the plasmid, we will reintroduce the plasmid into prepared bacterial cells. So, how do we physically separate the bacteria with our plasmid versus those that don't contain this plasmid? Remember that plasmids have antibiotic resistance, and well, you and I, being world-class scientists, showed tremendous foresight in our earlier stages of planning, so we simply introduce an antibiotic, hence preserving the bacteria with our plasmids while subsequently killing those without a plasmid. Now what? Glad you asked. We just leave the plasmid-bearing bacteria to its natural order of reproduction, and within days or even hours, it will have churned out large quantities of our target protein. And voila, insulin. Gene cloning is so easy, my grandma can do it. But her eyesight is failing, so we probably don't want her around expensive lab equipment. Apart from their use as factories of biomedical proteins, prokaryotes themselves are often home to potentially novel sources for new antibiotics. For example, in a recently discovered soil bacterium, a new antibiotic, teixobactin, was found to be an effective solution to MRSA, an infamous bacterial strain that is found in hospitals and known to be resistant to multiple antibiotics. While only about 16,000 prokaryotic species have been identified so far, a single handful of soil could contain 10,000 prokaryotic species by some estimate. Just imagine how many new antibiotic discoveries are waiting to be made in that handful of soil. And if you're even more of an optimist, the next cure to a disease like HIV is perhaps in that soil. The field of microbiology and its subsets have long fascinated me with its quantitative perspective on the living world. It is one of the most versatile fields of science and has endless applications to our society and its well-being, as seeing antibiotics or even beer brewing. I can see myself one day conducting research in a university laboratory, slowly chipping away at the surface of a grand discovery. I must ultimately thank the prokaryotes, whose approbation I've been struggling to win, for it was they who first sparked my interest in biology. My closing remarks would be that the diversity in life is elegant and at times breathtaking, or if you want to put it in Charles Darwin's words, endless forms most beautiful and most wonderful.